Let's check out capsid stability, aggregation, and titer with Uncle. I'm Kevin, and I'd like to thank the team that helped put this presentation together, Ross, Amrita, and Patrick. Uncle is an all-in-one multimodal stability platform built for biologics. It uses two lasers to excite either label-free protein intrinsic fluorescence or a variety of dye-based fluorescence. With full-spectrum fluorescence detection, Uncle can pick up both kinds of signals. Today we're going to be looking a lot more into Uncle's full spectrum of fluorescence with a fluorescent dye sensitive to DNA and by looking at the fluorescence of the native protein of AAV capsids. SLS is the scattered light from those two lasers, and it gives information on when aggregates form. DLS is the dynamic light scattering system and allows for detailed measurements of size and size distribution. These three detection methods can be operated isothermally at room temperature or across a heat ramp designed to thermally stress samples and see which ones take the heat. Uncle can run a thermal ramp on anywhere from 1 to 48 samples and only requires 9 microliters at a time. How it does this is with the uni. The uni is 16 coarse cuvettes held in an aluminum frame so you get even heating across the sample. Each well only takes 9 microliters of volume and it's simply pipetted into the uni cuvette. The uni is then sealed in the blue frame between two silicone seals so you don't have to worry about sample evaporation even as they heat up. This poster presentation will be looking at AAV capsid stability two ways. In one way, as proteins unfold and capsid disruption occurs, and in a second way, as DNA is leaking from relatively intact capsids. So on Uncle, we'll look at capsid disruption through intrinsic fluorescence, which is the protein fluorescence that will change as capsid proteins unfold. Here we have a quick look at the intrinsic fluorescence signal collected by Uncle on a sample of AAV9. Each line is a fluorescence read at a different temperature. Uncle will excite fluorescence with its 266 nanometer UV laser, where it will also collect SLS at that same wavelength. When that laser excites the AAV capsid proteins, they will fluoresce in this intrinsic fluorescence range between 300 and about 430 nanometers. As the temperature increases, the signal intensity drops and shifts to the right, called a redshift. The blue arrow is meant to guide the eye to the redshift, where the dashed blue line shows the true vertical. Our analysis will typically monitor the spectral center of mass under that fluorescence signal, which we'll call the barycentric mean, or BCM. As the signal shifts to the right, that BCM will increase. You can also note the SLS signals from both the 266 and the 473 nanometer lasers. Here we're looking at the analysis of the previous fluorescence data. Along the y-axis, we're tracking the intrinsic fluorescence BCM wavelength. You'll notice as temperature increases, the signal also increases, indicating a redshift. Where there's an inflection point in this curve, we've indicated a Tm, and where the curve starts to increase, we've indicated a T onset temperature. This data is shown for AAV at 1 × 10 to the 13th Bg per mil, but Tms are detectable for intrinsic fluorescence down to 5 × 10 to the 12th. We can also see the aggregation here in the SLS data in green, which tells us where aggregation starts with the T-ag. So in this case, you'll actually see that the T-ag indicates aggregation simultaneously with the onset of unfolding behavior. This is something that we've seen consistently across a lot of different serotypes, but the temperature which this occurs varies both based on serotype and buffer. If we want to compare those unfolding aggregation temperatures to the literature, we can look at this paper by Bennett, who used a dye-based method to look at protein unfolding. Comparing Uncle's results and the results shown by Bennett, we get very good agreement for 3 different serotypes tested here, AAVs 1, 2, and 9. Now let's take a look at the other stability pathway for AAV, the genome ejection pathway. To study this, we're going to add cybergold to our samples so that we'll know when DNA ejection is occurring. In this case, cybergold has very low fluorescence in the absence of DNA, but increases in fluorescence as DNA is released. To track this behavior using Uncle, we'll again use its full spectrum fluorescence. Uncle will excite cybergold using its 473 nanometer laser, and will follow its emission between 500 and 630 nanometers. Uncle will again use SLS, but this time with the 473 nanometer laser to track aggregation of samples during thermal ramps. Here's a look at the analysis for the cybergold fluorescence experiment. In green, we'll be looking at the fluorescence of the cybergold dye, and we're tracking the area under the curve. In blue, again, we have the SLS of the 473 nanometer laser. Starting at 25 degrees Celsius, we see some initial fluorescence signal from the cyber gold because we know that we have some free-floating DNA present in the sample. As temperature increases, cyber gold fluorescence decreases because we know that there's a relationship between temperature and cyber gold fluorescence. However, the headline of this experiment is starting at 50 degrees Celsius when we begin to see the cyber gold fluorescence signal increasing, indicating that genome ejection is beginning to occur from our AAV capsid. We've also identified a melting temperature at 58 degrees Celsius, which is what we'll use to compare different genome ejection curves across serotypes and different formulation buffers. This genome ejection behavior continues until approximately 75 degrees Celsius, where again we see our SLS signal spike, indicating that aggregation is occurring in our sample. Here we're looking at that genome ejection behavior for two different serotypes, and we're again looking at cyber gold fluorescence. For AAV9, we're seeing melting temperatures around 57 degrees Celsius, and for AAV2, we're seeing melting temperature 8 degrees lower at 49.6 degrees Celsius. This indicates that genome ejection is occurring much earlier for AAV2. If we're looking at the bottom of the slide, we can compare the unfolding behavior by intrinsic fluorescence for the two serotypes, shown in the first column, which again shows us that AAV2 is less stable and has a lower unfolding temperature. Compare that to the genome ejection behavior and the aggregation behavior shown with TAG at 473 nanometers. The difference between the two serotypes changes for all three metrics. For protein unfolding, for genome injection, and for aggregation behavior, so you can't simply measure one and extrapolate to the others. It's important to track all three of these behaviors. So what we've seen from these data is that AAV has two different stability pathways that happens at two different temperatures. Capsid stability can be identified by intrinsic fluorescence and aggregation metrics and tends to happen above 70 degrees Celsius. Genome ejection is an independent pathway that happens as DNA is ejected from relatively intact capsids and can be monitored using DNA-sensitive dyes like cybergold. It is important to measure both of these pathways to fully understand the behavior of your AAV. Using Uncle's new vial toolbox application, we can readily report the initial fluorescence area signal related to the signal from cybergold, and that can tell us information about the free-floating DNA in our sample. We can also look at the cybergold signal after a thermal ramp when we come back down to 25 degrees Celsius again and take a final read on fluorescence of cybergold. This is useful for measuring the final concentration of DNA in your sample after all the capsids have ruptured. So with one experiment, we'll get a picture of both the initial free-floating DNA and the amount of DNA being detected by cybergold at the end of the experiment, expressed as how much fluorescence the cybergold is emitting. Uncle's DLS is also very useful for AAV samples, where in this case we're looking at DLS from before and after a thermal ramp experiment. At 25 degrees Celsius, our AAV diameter is about 25 to 30 nanometers, which is great and what we expect for an intact and monodispersed AAV capsid. After the thermal ramp, at 95 degrees Celsius, we see aggregation, which is what we expect and what the SLS indicated for this sample as well. So this is great because we're able to confirm that we're starting with a sample that's in good shape and that we can rely on the thermal ramp results as an experiment that was performed on a single non-aggregated sample of AAV. And if we want to extract from that DLS experiment a metric called particle intensity, that can help us understand how many capsids are present in the sample. So we're reporting the particle intensity on the y-axis in the left, and we know that we can build a standard curve against that metric related to the AAV concentration. Then we can use that standard curve for unknown samples shown on the right, and therefore calculate AAV concentration. Because the slope of the graph on the right is close to 1 and the R-squared is above 0.99, we know that this is a very linear metric that correlates very well with concentration of particles. So let's look at how we can use Uncle to look at more AAV stability behavior. Using Uncle's two lasers and full spectrum fluorescence, we're able to look at both pathways of AAV stability, genome ejection, and capsid disruption. DLS is able to look at sample quality before a thermal ramp and is another quantitative measure of aggregation. And its particle intensity and cybergold fluorescence metrics are useful tools to look at capsid count and DNA on the same sample. And importantly, most of this information actually is available in only one experiment using only one 9 microliter sample volume. So if you have questions, want to learn more about our Uncle system, or just want to quote, please don't hesitate to visit our website or email info@unchainedlabs.com.